Hi, it's still Friday, and it's still my period 3 class. Say hi.
Hi.
And we're moving on to translation now. So we've already gone over the steps of how we make our RNA from our DNA, and now we want to change this into a new language. Instead of a nucleic acid language, we're going to turn it into a protein language. So in order to do that, we want to remember that every 3 bases on the mRNA is called a codon. Okay, and the tRNAs are going to land on the codons and start hooking the amino acids together to build the protein. Now, I want to jump to the very end, and then I'll come back and film the details and show you a little animation because I think it will help you. We watch that now. Maybe. Okay. We'll go back and look at the details, but we'll watch this first.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has 2 sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. The initiating transfer RNA, carrying its amino acid, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of 1 codon and...
Okay, now, in your mind, you know, you can look at a picture and it looks one way if you look at it one way, right? Okay, when you see the ribosome, a lot of times people want to think of the red mRNA is moving, but what you really know is moving is what? The ribosome is moving. Now they have shown you 2 spots here, the A site and the P site. There's a 3rd spot there that they're oversimplifying here, and that's the E site, and E is for exit. And so what you want to envision, you can name it the way they did, but another way of naming it is you have the amino acid site and the protein site. So you have 2 tRNAs landing side by side. The new guy who was in the A site, the amino acid site, he only has 1 thing on him, right? Okay, as the ribosome shifts, he's going to move the guy that was in the A spot and put him into the P spot. And in the shifting process, this ribosome, and we know, right, that most enzymes are made out of proteins, but some can be made out of RNA. This is a ribozyme. It's going to catalyze the reaction to join the amino acids together. And the new guy gets dumped on, and a peptide bond is formed between his amino acid and the whole chain of amino acids. Okay? And now he's in the P spot, and the guy that was in the P spot is in the E spot, exit, and he's going to what? Leave. And along comes the next tRNA, and he's going to land in the what spot? A. And then as the ribosome shifts, he's going to move into the what? P, and he's getting dumped on. And so on and so on and so on until the codon says stop. Okay? So watch for that.
And the first transfer RNA is released.
E spot gone. Oh.
A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of 1 codon. This is followed by the 2 amino acids on the transfer RNA in the P site being transferred to the 3rd amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Okay, I want you to ping pong back and forth. Youngest, go first. You're going to go tell him 2 things. This is what I know for sure about translation. Okay, content-wise. Go back and forth twice. Go. This is what I know for sure. Just one statement that you know for sure. Okay, we good? All right, so let's walk through that. This ribosome is drawn inordinately small here, okay? But the spot right here on the ribosome that's holding this particular tRNA with its long amino acid, what spot would that be called? P. And this spot right here on the ribosome that's holding just the single amino acid would be what? The A. And the spot that this guy just left from on here would be called the E site. All right? Okay. So this is why you can have several ribosomes moving along here, each putting that particular codon anticodon complex, right, in the different spots, building several proteins off one mRNA. So we know that every 3 bases is called a codon, and if your codon was CGA, what would be the amino acid that the tRNA brings you? If your codon is CGA, if you can read up here, it might be too far. Okay, so my first base is C, so it could be anything here at this point. But then it says my second one is what? G. So these are all out of the running, and now I'm here. And then it doesn't even matter what the third one is because it's still going to be what each time? Arginine. Okay, so if that's my codon, then my amino acid that the tRNA is going to bring me is arginine. Yes?
So this chart only shows codons, not the anticodons?
Right. This right here is the mRNA code. And you know how you're going to remember that? It's the what? You're reading the message, right? You read the letter that is coming from the DNA. This is in the mRNA. Yes?
You would be able to figure out the anticodon, though.
I certainly would. Let's figure out that anticodon of this amino acid was brought here. Let's figure out what the tRNA would be that brought that arginine. What would be the letters that we have right there? G, C, U. You'd totally be able to figure it out. Okay? And this is what the tRNA looks like. Yes, it is single-stranded, but what you have is you have these loops, these different loops. And this loop right here is the anticodon that it's having. And then look, loop, 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 and then here, it's the 3' end that has the amino acid. Why do we have 3 and 5? Because tRNA, just like mRNA, was transcribed from the DNA. And you read from 3 to 5, making 5 to 3. On its 3' end is where it puts its amino acid. Now there's an enzyme that just puts those amino acids on there. Okay, the right amino acid on the right tRNA. Questions, concerns? All right, let's keep going. So if this is the anticodon, you are going to put in your click for me what the codon is. This would be, read it left to right, top to bottom. What? What does it say? Go Emperor, Java. Okay. So, good Lord. G with C, C with G, U with A. Look at your BioBuddies clicker right now.
Wait.
My father did think that.
You asked the question, I was sitting him. You're thinking. No, it is. Remember, if you need attention, get a hug before you come to school so you don't annoy us. I'm just saying. So glad that got recorded. Such a love. This is not difficult. And if it is difficult, you need to do some better preparation for this class. You can do it, Scarecrow, or not. Oh. Okay, people. If the mRNA is this, okay, what is complementary to this mRNA? What would the DNA have to be? G, C with G, then what? C, C. Okay, G, C, C. Could you please, because 43, oh, maybe a little bit more, because some of you didn't capitalize because you didn't know what uppercase meant. That's a shocking amount to miss. So could you please check with your BioBuddy right now and be honest engine, show them your clicker, let's have a discussion. At this point, you shouldn't be missing these. Hey, so what that's saying is if I'm out here and here's my codon, and my codon reads CGG, then what was the DNA that made it? If it reads CGG, then it must have been GCC, because G with C, right? And then C with G and C with G. Yes?
So the anticodon will pretty much be what the DNA code for originally, unless you're using like A's and U's.
Well, the anticodon and codons will always have U's, but yeah, what you have in the DNA, you're going to see in the tRNA. But you got to. What you don't want to do is you don't want to run into trouble that way. You want to imagine, where am I in this process? Yeah. Always imagine that big picture. I'm the mRNA, so the DNA was this. So the tRNA is this. Okay? Yeah. Okay, ask your BioBuddy, are you still in that Shadowlands, the 57% of you who struggled? Are you in the sunshine yet? Ask them. Anybody want to come to the sunshine? Do you need me to reach out a hand and bring you out of the shadows? What can I go over? Come to the light. Come to the light. Yes.
I thought when you said that you wanted to see like which amino acid was produced.
Wait, I can't hear. Shh. Go.
So I thought that when you said you wanted to see which amino acid was produced, you would look at the actual codon, not the tRNA.
You do. You only look at the codon. But what I asked here is I said, what was the DNA, the sequence of bases, that coded for this amino acid? So here's the amino acid. I wanted to know, what must the DNA have been so that you ended up with arginine? Does that make sense now? Now, it doesn't go DNA directly to amino acid, right? It goes DNA transcribed into mRNA, use the mRNA to build a protein, and which arginine would be a part of, right? Okay, but I want to know what the DNA was that dictated that arginine was there. Okay? What if the DNA is messed up? Then you wouldn't get arginine, would you? You'd get something else. And what do we know about protein structure? If I mess up the primary structure, I'll probably still get the secondary structure, which is an alpha helix or a beta pleated sheet, because that's due to the carboxyl and the amino groups, right? But where am I going to run into trouble? Tertiary structure. I'm going to get some misfolds here, which could then influence my, what's the next structure? Quaternary. So if your DNA is messed up, so too will be your proteins. That's bad. What are some ways you can mess up your DNA? Yes. What? Carcinogens. So don't expose your body to carcinogens. What's another way your DNA can be messed up? Inheritance. So don't expose, no. So knowing that, if that's why they ask you your family history when you go to the doctor. Because you want to give them as much family history as you have to explain that. But what you're going to learn in chapter 13 is there are some cancers and diseases you can get that don't have anything to do with your DNA. But remember how we talked about supercoiling, how we put DNA away? There's things called methylation. Okay, where remember the histone proteins that it's logged on, or the DNA itself, depending on if you put a methyl group on there, will either make it unwind and expose it or keep it tightly coiled and put away. So if it's an important gene and it's tightly coiled and put away, doesn't mean that the gene wasn't there, that your mom or your dad didn't give it to you. You just don't have any what to it? Access to it. Okay, they gave you a safe with $1 million, but you don't have the key to get it out. Okay? That's called epigenetic inheritance. Epi means what? Above. Above the genes. Okay, so we're going to learn about that as well. All right, moving on. Okay? Remember how we talked about there are 64 different combinations we could come up with. We have 4 different bases that could be in 3 different spots, right? 4 different bases in 3 different spots. So you could have 64 different combinations. We only have 61 because 3 of those are what? Stop codons. But so we had to do that, or your body had to do that, because you wouldn't have enough options if you only had 2 bases in your codon. You needed 3. So we've overdone it, right? There are really, as far as tRNAs go, there are not 61 different tRNAs. And that has to do with this Wobble hypothesis. Remember how we saw with the arginine that there are multiple ways to get it, right? And so as a result of that, you don't have to have every tRNA represented, and we don't. And then sometimes that pairing relationship doesn't even have to be perfect. And sometimes you can have a group that's attached onto it right here that can change it from one type of base actually into another, and that's called the Wobble hypothesis. And I'm going to pause just for a minute and look at your notes and get caught up for just a second. All right. So, next. I taught you that you had to get the right amino acid on, and you have an enzyme for every one of those. Again, the importance of those enzymes, right? And this is one because you're joining. Is that anabolic or catabolic? Anabolic's about building, right? In this case, it's requiring ATP in order to do that. Every single time you add an amino acid onto your tRNAs to rebuild and make that tRNA functional again, it does cost you an ATP. Another important detail, there is both a small and a large ribosomal subunit. And when that mRNA is made and transcribed, and for us in eukaryotic cells, it has to get capped and tailed and snurped. When all of that is done, it will hook up, and it leaves the nucleus. It hooks up with the small ribosomal subunit and then the large ribosomal subunit. And it's like all in there. Now it's ready to build a protein within that safe zone of a small and large ribosomal subunit. And there are 3 spots in there, right? Youngest BioBuddy, tell the oldest BioBuddy, what are the 3 spots? Go ahead, inside the ribosome.
I got it done.
Okay, and the large ribosomal subunit is made out of 3 RNAs, and the small ribosomal subunit is made out of 1. Do you remember when we were studying the 3 domains? What are the 3 domains? Eukarya, bacteria, and Archaea. And remember I talked to you about how they used to think Archaea was really old, hence the name Archaea, like ancient, but really it's what? Newer. Its process of translation and its ribosomes are more similar to the eukaryotic ribosomes than the prokaryotic ribosomes. So this is where that would come into play here. Now, take a look at this picture, and oldest bio buddy, this is yours to explain. Tell what's happening in this. They're not showing you the E spot, but there is one right there. Go ahead.
Now, the first tRNA it's not like
Okay, clarifying questions on this picture.
It's a magic star.
All right. All right. Now, as we have also learned before, that proteins are not functional until they're actually put into their right shape. That tertiary structure, that folding, proteins will assist in that. If that doesn't happen, if it's not folded correctly, then you need to get rid of it. But the chaperone proteins will fold them into their appropriate shape. This is called a polyribosome. Now, before, where we were looking, remember the yellow strand of DNA, and I said, okay, RNA polymerase, which is the 3 and the 5. That was transcription. Now we are looking at a picture of translation. Now we're using the RNA, and now the things that are dangling off are not mRNA. The things that are dangling off like a mouse's tail here are actually the proteins. So this ribosome just got here. This ribosome's been here for a while. And so he has a very long polypeptide or a long protein. And a polyribosome is all of this mRNA, this one strand of mRNA is getting read by several, like doc, ribosomes keep docking onto it, reading it, building, and making lots of this particular protein, which the cell must need. Now, what could block it from doing this? Well, enzymes could start to break down that RNA. Enzymes could start to break down that RNA, or you could end up getting a small piece of RNA that binds on here to make it double-stranded. Now the ribosome, when it gets to that spot, and it won't be able to translate it. So that could be a control mechanism as well. Okay, youngest, explain this picture, right? No. Oldest? Oh, youngest, this is your picture. Go ahead. You can do it. I'm trying to give you opportunities. Teach it to them. And I'm adding things to it. You explain what I add on. Environmental Protection Agency. All right, now let's watch. Questions you want to ask me about that picture? Yes. What are the what? Okay, so in this diagram, they really didn't show you, but there would be like a notch right here that would be the A spot, and then here, right here would be the P spot, and then there would be an E spot right here. Okay, you know what? You'll be able to see it better in this picture. All right, so the mRNA first hooks up with the small ribosomal subunit. Then the large ribosomal subunit will dock. It won't dock on until the mRNA and the small ribosomal subunit are hooked up. Now we are seeing it's like it's cut away and we're looking to the interior. This would normally be blocked. The very, very first tRNA will land at this P spot right here. And I want you to see right here what it says. Start codon. What are the first three letters? A, U, G. Now, I want you to look here at this chart. Find AUG. Same chart, just written a little bit different. Do you see it? What amino acid does it carry? Methionine. Okay? This right here is the only way to get this amino acid. This is what's called the initiation or start codon. Every single protein always starts with the first amino acid being methionine. Now, as it's folded and put in its proper form, it might be clipped out. But initially, that initial chain, that primary structure of every single protein, the primary structure of every single protein starts with methionine. This lets you know where it needs to start reading, where the codon needs to snap on. Where the ribosome needs to snap on is always right here. And it puts it right here in the P spot. Okay, so the first one only lands right here. Then the next tRNA is going to land right here. And once he lands here, this ribosome is going to shift in this direction. Okay, and this methionine is going to get dumped on what he is carrying. And as the ribosome shifts to here, then this one will be in the P spot, and this one will be in the what? E spot. Okay, so here it's been going on for a while. Here's the new guy. He's coming into what spot? A. And I always think A amino acid. It's only got one. P proteins. It's going to land here, and as the ribosome shifts, then this long chain is going to be put on here. Look, what is the very first one? Methionine. And it will be on every single protein. And this is going to continue and continue and go on. And this is your phenotype, right? The DNA that this mRNA came from, this is your genotype. Okay, you transcribe and then translate in order to get your protein, which is your phenotype. Yes? Okay, and if you just have one gene for tall, you will be what? Tall if you're a pea plant. Right? Okay, because we have 2 copies of every single genotype, except for males, you only have one X. And whatever you get, right? So put it together. When we say, I have blue eyes, you actually have an absence of pigment, which means that gene is not getting laid down, which is you're seeing the black of your retina come through. But if you have brown eyes, you're coding for a pigment. You're making that brown protein that gets in your eye in order to protect your eye. Yes? And other genes can modify that. You can get some other pigments and some sparkles and little yellows and outlines. Right? That's your phenotype that was coded for from your genotype. Okay? And this will continue until you reach your stop codon. And then it's just going to break apart. And here's your protein. And there's a small and large that can get reused again. We already know how to do that, so we're not going to do that again. There's another chart. There are, now this one kind of puts it all together from what you learned. Can I get eyes up here? Up at the top, you can see transcription. Wow. So here's the DNA, here's the mRNA. At this point, it has introns and exons, but then we need to process it, and you're going to take out the introns and just leave the exons. You have a cap and a tail, hooking up with small and then large ribosomal subunit. You know the dance that's going on right here, the translation dance. This ribosome, if it's on ER, this ribosome is called what? If the ribosome is on the ER, that ER is called rough ER, right? And now that protein that you just made is moving into the endoplasmic reticulum, and then it could get shipped to the Golgi, who modifies it and packages it for release, either kept inside the cell or out. Put it all together. Okay? Put it all together. This is your rough ER. Okay? Rough ER. We know we have a leading and a lagging. We already know all of that. Explain this, young one. Go ahead, young one. Explain this one. You should be able to do this. We all right? Okay, keep going. When DNA goes bad, then if the genotype is bad, then the phenotype is bad. And we now know why, because we know how the gene gets translated into a protein, right? Transcribed and then translated. So a mutation is a permanent change in the sequence of DNA bases. We're going to talk more about mutations in chapter 13 when we also talk about the control. Okay, and we already know how to do this. I don't know why I have 1,000 of those in there. That's the end. Yay!